Hi guys, so previous 4 videos we have learned the DNA profiling. So it's a very detailed procedure. Now we're going to check here how the questions come in the exams. Now this is a one question. Explain how gel electrophoresis can be used to determine the father of the cub. So that's a captive breeding programs. Captive breeding program, the conservation method. We need to find it out the father of the cub, because this will prevent inbreeding depressions, because inbreeding is not really a good thing in a captive breeding programs. However, we want to minimize the inbreeding, so we want to maintain the records. So, we maintain the record books of all the animals and how their father, mother, and the pedigrees and everything. So here, if we miss something that the father of this cub, so we want to find it out the father of that cub using the gel electrophoresis. That's a 4 marks question. So if you want to get 4 marks, complete mark, so all the videos what you have learned, we have to write now quite a good answer to get it. Number 1, how we get the DNA. So practically we can get the DNA from without harming the animals. It could be from the hair. It could be from the blood. It could be from the saliva, or maybe sperm. It's available if a sperm is available from father. And this DNA, again, we have to get it from a cub, a father, fathers, and the mother. That's the source of DNA. So source of DNA, so it's a number 1, the hair, blood, saliva, and sperm. And then from whom we get the DNA could be cub, fathers, because more than one father, and the mother. Because here, like a paternity testing, we know the cub, we know the mother, and we don't know the father. So it's just like a paternity testing of animals now. Then after that, what you do, we learn the gel electrophoresis method. In gel electrophoresis, we use a restriction endonucleases to cut the DNA into fragments. So we have learned short tandem repeats and variable number tandem repeats and restriction site, that's a palindrome site, and how the DNA is cut into a different length of the bands. And after that, what we do, we can load the DNA fragments into the gel and apply the current, that's the current, and to run the gel. So we place the gel and they apply the DNA sample prepared, and we run the gels. So then you can mention, if you want, the 4th point, we can perform the southern blotting. That means bands you transfer into a nitrocellulose paper and use suitable complementary sequence to visualize the bands. And after that, the final stage, you have these bands like this way. That's a father, mother, and the cub, and maybe several fathers. So you compare the position of the gel bands to find which one is the right father. So all the details you can find in the video. However, here in a question to get a 4 marks, you need to highlight this one, restriction endonucleases, and the running the gels, southern blot, and they can have at least 1 marking point. That's a second marking point, and third marking point, and fourth marking point, and fifth marking point, and sixth marking point. So you can have at least a 6 marking points to get 4 marks for your answer. So if you run a good the method of the art of writing these whole points, you can achieve 4 marks by writing these questions. So all the details and learning for that one previous video, you can check it out. Thank you very much.